All right, number 1 says you should be explaining the differences between RNA and DNA. So, what are some differences? Cedric, you know any differences between RNA and DNA? Okay, so in RNA, let's do like this. RNA, we use uracil. In DNA, it's thymine. That would be one difference. What else? Okay, both on top of it. DNA is a double helix. RNA is a single. Okay, they both have that helical structure. They both twist. RNA complementary. Will be complementary to DNA it's built on. Okay, what else? Okay, DNA stays in the nucleus. So the nucleus only. The RNA goes from the nucleus to the ribosome. This messenger RNA. Okay, any other differences? Okay, so the sugar that's involved. So DNA is the deoxy, whereas the RNA is the regular ribose. Okay, so those are some things you should be able to compare and contrast between those two different molecules. But we also have different types of RNA. You should know what each of those are, and what their function is. Oh, okay, Jason. Right. I understand. Okay, so the messenger RNA, remember, that's the one that actually takes the code to the ribosome. That's the one that we've talked most about. It's involved in both the process of transcription and translation, whereas the other two types of RNA will not be involved in transcription at all. They're only involved in translation. The transfer RNA, of course, is the thing that's going to bring the amino acids to the mRNA. So that's the thing that's actually carrying the amino acids along, and it's able to connect to the messenger RNA using the to build that protein strand. What about the ribosomal RNA? It's basically the structure of the ribosome. So, I mean, yeah, you could kind of say because it's part of the ribosome that it makes proteins. But this is basically the structure of ribosome. But our ribosome is only made of ribosomal RNA. What else is it made of? Okay. Protein. So ribosomes, remember, are ribosomal RNA and protein all wound together. These two things make up the actual ribosome. Taste the code. Okay, we should be able to describe the overall process of transcription. So, what's the product of transcription? What do we make? Which type of RNA? Right, the messengers. So the whole purpose of transcription is to make the messenger RNA. That's what transcription is. It has to be made based on the code of the DNA. But the whole point of transcription is to make messenger RNA. That's what transcription is all about. Okay, so here the actual process, we have that RNA polymerase, which is going to build the messenger RNA using DNA as a template. So you remember that ribosomal RNA unwinds the DNA in one little section. It reads starting at the start codon, along builds the messenger RNA, one piece at a time, one at a time. The T's on the R or the DNA and all those other things, making sure everything gives the code where it's going to match with an amine on the DNA molecule. So using that template of the DNA. Okay, now is the messenger RNA strand going to stretch the entire length of a chromosome? No, not even close. These things are built in tiny little sections, little pieces of messenger RNA in between here and there along that chromosome, along that DNA strand. So start at those start codons, stop codons. Okay. Now, number 4 says you should also be able to describe the overall process of translation. So, what are we making in translation? No. Amino acids are already made. We're putting together the amino acids to make proteins. Okay? So be careful about that. Right, they're made out of, but I mean, we're not making amino acids. Let's say we're making an amino acid chain. But the whole point of translation is to, well, let's see, read messenger RNA to make protein. Talk about the ins and outs of that a little bit further down. The whole point is that the ribosome, that codon of 3 base pairs together, and then transfer RNA and match itself. It's complementary to those. It's going to come up and match itself up to the messenger RNA. All right, number 5 says you should be able to transcribe a given DNA strand to the correct messenger RNA. So I have a bunch of A's and G's and C's and T's on there. You should be able to match up the correct sequence. Yeah, the only tricky part, if it is a tricky part, is you remember messenger RNA match used no thymines. Okay. Number 6 says you should know what a codon and an anticodon are and where you would find them. So both of these actually are groups of 3 nucleotides in a row. what would be the difference between codon and anticodon? Anticodon is found on the tRNA. Where's the codon found? On the messenger RNA. Okay. They're both groups of 3 nucleotides. Okay. An anticodon is going to be nucleotides on the transfer. But it has to be complementary to a codon. So if we've got a codon that's AU, this one would have to be AA. It has to be complementary so that they can match. Right. So the whole point, the way the code gets passed on from one thing to another is these complementary things. So the messenger RNA is to be complementary to the template DNA that it was built from. It's also the transfer RNA anticodons have to be complementary to the right amino acid. So yeah, we could go back and look at the DNA strand that it was built from and know that on the DNA strand, the 3 sequence of 3 nucleotides there would be TAA. So, yeah, the anticodon and the strand on the chunk on the DNA are basically the same sequence. Okay, number 7 says you should be able to use a codon chart to predict the sequence of amino acids based on a strand of DNA or messenger RNA. Yeah, we've done enough of that. I will have the charts that I handed out for you Monday. If other one's better, it's fine. Just make sure you have copies of it. You're writing on it. That's fine. Just okay. Biggest thing here. Remember, the charts, both of those charts, are based on the messenger RNA strand. So if I give you a chunk of DNA, if I say this is a DNA strand, or heck, I shouldn't even have to tell you because if you see T's in there, you know it has to be a T. I say what amino acids are going to come out of this. You have to transcribe that first. Okay? You have to write down the complementary messenger RNA strand to be able to find what those sequence of amino acids are, because the messenger RNA, I should say the codon chart, is based on that messenger RNA strand. So be careful about that. If you're given the DNA, make sure you make the messenger RNA before you feed the codon. I may ask you if I give you a codon. But in a specific chromosome, okay, transfer RNA is going, not transfer, sorry, the RNA polymerase is going to attach to one side where it has the correct start sequence. Okay. Now, as far as for you guys to know, I'm just going to give you one side. I wouldn't give you 2 sides. Or if I did, then I'd be I'll just give you one side. As far as physically what happens, on one side you're going to have that start sequence, on the other side because it's not number 8 says you should be able to find the special codons for starting or stopping the transcription process. So that's just about reading your chart correctly. Recognize, hey, this means stop. This one here is the start codon. There's only one start codon. There are 3 different stop codons. Okay. What's that? Yes, that's the stop. Yes, that's specifically the stop. Each one of those. Okay. Number 9 says describe the different sites of a ribosome and what each is for. Okay. So we talked about the different sites. To help you remember it, it's spelled backwards. Yeah. Yes. So the actual letters, well, we just use well, sort of. Uh, as far as specific words that they stand for, it has to do with the way the chemistry goes together. So that's not necessarily makes sense to you, but we can use words that kind of sound like or that use those letters to help us. So A is where the attachment of uh, uh, um, the transfer RNA to the messenger RNA happens. Okay. Yes, I don't know what was wrong. So A doesn't actually stand for attachment, but we can use that word. You need to know what is happening. So A is where the transfer RNA meets up with the messenger. Place on the ribosome. Those two things. Yeah, where tRNA, the anticodon, attaches or meets up with the messenger RNA. The P site we can think of as production. Somebody can think of some other keyword. Harry. How about a P that actually makes sense with what's going on? Okay, I think of it as production because this is where one amino acid, from this, the whole thing moves along. So the transfer RNA is attached. When it moves along to this site, that's where the amino acid on that one transfer RNA gets added to the growing chain. So in the production kind of site, think of it as production site, that's where we have adding of amino acids. And we call that amino acid chain growing protein. Okay. So remember the way it works is RNAs here. This is an A site. This one's in the B site. This one's in the A site. When it moves along, and it comes over, and then this gets transferred to the amino acid sticking on this one. So how much chains up there, and then this one can go away, and then this one will go on, and transfer gets changed to the next amino acid. So it takes the whole chain and transfers it to the next amino acid at the B site. Then E we can think of as exit. That's where the empty transfer RNA attaches to a messenger RNA. Okay, that's a good question. In most diagrams where they show this happening, they're going to lay out the messenger RNA. The whole right to left. You know what I'm saying? So, like, if this is the ribosome and this is the have something here. Here we go. Oh, that cord is attached to something. So there, if this is the ribosome here, my fist is the ribosome. The way the messenger RNA in diagrams is always set up is it's full and it's kind of responds in this direction. No, because this is where the A site is where the transfer RNA comes in. And so the ribosome itself doesn't move along, right? The ribosome sits in place. It's actually the messenger. It moves along so that things go along. So, yes, you're right. You could think of it in the opposite direction. There's no reason not to, except that it's if you put it in I'm not going to ask you for the place. You just need to be able to describe. Okay, so yeah, you're right. The order that we learn these in, as far as putting them down that way, it just has to do with the diagrams. Okay, number 10 says you should be able to describe the basic structure and function of an operon. This, remember, only in bacteria. Right? It's a simple way of gene regulation. So we have the I can't think today. Thank you. We have the start sequence, which is called the promoter. That's the word that I'm looking for. Promoter. Okay, the place where the thing is actually going to start. We have the gene that is actually going to be read. Okay, then we have the operator. Thank you. I may have a tumor. Repressor, yes. Okay, so remember, first three things are actually on the DNA sequence, right? They're sections of DNA. So the gene is the thing that we actually want. The promoter is where the RNA polymerase can attach. The operator is the place where the repressor can attach. If the repressor attaches, then it blocks the RNA polymerase from attaching at the promoter, and transcription is not going to happen. So a lot of times this happens when, hey, the bacteria cell has enough of this type of protein. We don't want to make a whole bunch of extra that we can't use. So that's some sort of stimulation is going to cause the repressor to go attach to the DNA strand, and transcription can't happen. Can't happen. Okay, now different ways. In some cases, an enzyme will actually basically break it down. Okay. In some cases, the attachment of another molecule will change it off. So a few different ways. Removal of the repressor, and then you'll let transcription be able to go. Okay. So that's the basic stuff that you need to know there. Number 11, compare and contrast gene regulation in prokaryotes and eukaryotes. The basic thing you don't need to know here is that in eukaryotes, it's complicated. So this is prokaryotes, right? That's fairly easy to understand. One thing can block the RNA polymerase from attaching. In eukaryotes, there's lots of different things that happen. I'm not going to go too far in depth that, so just realize that it's more complicated.
It's like your relationship on Facebook. No. It's like a relationship status on Facebook.
No? I don't think you're going to dig yourself out of that hole. Okay. Real quick here, we don't have a whole lot of time left. Number 12 says you should be able to compare and contrast introns and exons. Okay? Introns are trash, right? We don't really want to call them trash because we know they do serve some functions now as far as how the exons are actually expressed. Basically, the introns are not sent to the ribosome. Okay? Now be careful here. They're still transcribed. So they're still put into the messenger RNA. Before the messenger RNA leaves, they're cut back out again. I know that seems like a wasteful process, but technology is much simpler. Sometimes things would be the easiest. So the messenger RNA still gets built with all the introns in there. Then it comes and it sticks out the exons, puts them all together, and takes those. Exons, remember, are the parts where we actually have the genes. Okay? Like we learned on the video, that doesn't necessarily mean that the introns are worthless. They do help decide how the exons get sent out and sent on. But they don't actually make it. Okay, number 13, list the different types of mutations and how each will affect the final protein. So, the basic thing here, we have what are called point mutations, and then we have frame shift mutations. Frame shift mutations, remember we have substitutions and deletions. Remember from the activity we did yesterday, those cause crazy changes because codons are always read 3 at a time. So if you take one out or you put one in, you pick up the whole sequence. Oh, I'm sorry. Getting in a hurry here. Not substitute, insertions and deletions, right? Those are the frame shift. Completely change the whole order that the codons are. Okay. Sorry. I should have done this one first. I got my line crossed here. Point mutations are going to be the substitutions. There's a couple different kinds. Silent ones, remember, don't actually change the amino acid sequence, right? It changes the code, but because of that whole redundancy thing, we're talking about number 14 right now too, because some amino acids can be coded by lots of different codes, then it doesn't actually change anything, and so we don't actually see that. So it has changed the actual code of the DNA, but you get the exact same protein. We call that a silent mutation. Okay. Then the other type of substitution, a specific one that we talked about was what's called the nonsense. What happens when you have a nonsense mutation? Stop. You get stop codon. We do have CRT in here the next couple of days. Here's something that's went over real quick. You can understand how to deal with that. Okay. Here's a question of the day. As we'll do that.